some of the cells and stain them so we could fluorescent sort them. Are we sorting some or are we just
flow cytometry, just reading?
Awesome. Okay, so we're just gonna do exactly. So we're gonna do just reading in order to probe if the thing worked at all, right? How many cells do we want to look at?
I think if we can get like 200,000 cells as a sample, take that out and stain it, that would be great.
Cool. Am I in the camera?
Yeah.
Okay. Alright. Look at us. Education bloggers. Cool. Okay, so I'm gonna pick up the 6 plates.
So yeah, you need to turn on the hood first because it takes some time for it to normalize. So you don't have to press anything; it's always on.
Got it.
So what you need to do is just like lift up the sash, all the way to here. Yeah, a little bit more. Yeah, and then it keep beeping because sometimes it doesn't recognize the airflow, so you pull it towards you a little bit,
Okay. Got it.
and it starts to recognize that it's up, and it will detect the airflow, and once it goes green, you're ready to use it, and you can turn the light again.
Yeah. And last time we turned on the UV,
Only after you finish working.
and does it automatically turn off?
It turns itself off automatically
That's
after
why.
an hour.
That's why I was wondering if we wanna, if we need to turn off anything else. I think in the streets lab it's like manual, like you have to always turn it on now. But that's cool.
Okay. Yeah.
Okay.
And so while it's setting up, we can take a look at the cells. So I can show you what I did with the cells yesterday because I have to change them out of the transaction fluid.
To the
And now this is-
antibiotics.
Yeah, right now it has antibiotics. And also that I saw that they are very dense. Like,
Dense?
there's a lot of cells in small area. I added more, so from like one well, which is these two that we electroporated. I added, I resuspended the cells in 9 mils of culture media so that you can have like 3 wells.
Awesome.
So that just give them more space to grow.
Awesome.
Otherwise, they may run out of nutrients.
Awesome.
So,
Cool.
Okay, so what I want you to do is actually to take cell count reading so that we know how much.
Yeah, I was
to
just gonna
take.
ask if you know the cell counts. Yeah.
So turn on the microscope and let's just take a look and see about the cell health, just to check there's no infection or anything. The lowest magnification.
Can I see?
Yeah, so I'm just planting a few.
I haven't really used that actually, so.
Yeah, so
I don't know
if you
how
look
close it
down
is.
the microscope and you can turn the focus in and out.
With that one, or are they both the same?
So this one is coarse, meaning that it has more turning power, and then this one is fine tuning. So you can just adjust focus with your eyes. So you should see little balls of cells, and those are the PFS.
Are the left and right side the same?
It's the same. And because I'm wearing glasses,
Yeah.
the magnification, like the depth is different. So
Yeah. I see. Yeah, there are many cells.
yeah, so that's a lot of cells.
Yeah.
So we should figure out like how much. I mean, like they are treated the same way. So if we count one well, it should be representative
I see.
to all of the cells, especially they are suspension
And how
cells.
did you move that? Can I try to
So
move it?
yeah, so you can move it using this knob here. So if you do this one, it's up and down. You can see how the platform moves.
Right.
And then the lower one is left to right.
Right.
Okay. Cool.
Ooh. And then they kind of shake.
Yeah.
Oh, so there are some that are like stuck to the plate.
Yeah, so sometimes
And then the top
they
one
just
is kind of shaking.
settle
That makes sense.
into the bottom. So that's why, like, when you count the cells, make sure you pipette up and down using a P1000 a few times. I will show you, and then I will
Awesome.
do one here, and then you do that one
Awesome.
there. Sound
Awesome.
good?
Yeah, you will do the ZFP off and I'll do the empty.
Yeah.
Perfect.
Okay. All right. So now we looked at them. There isn't anything weird floating around.
And you
That's
just
good.
placed it on top. You didn't like fix it anyhow, right?
No, so yeah, when you put it on, like there's
Oh,
this
I see,
thing
now I
that
see.
you can open
Yeah.
it, so that would like clip it in place, so it
Yeah.
doesn't move around
Great,
when
great.
you are.
Yeah, that's what I didn't
looking
see
at it.
before. Perfect.
Yeah. Okay.
Awesome. And then you turn it off. Yeah. Everywhere.
Yeah.
This one.
So it's this one here. So
Perfect.
when I don't use it, I turn it off.
Perfect.
And I like doing my cell staining in these V bottom plates. So what I'm gonna do is put this one inside for me to use later. And so one thing we could do is to just spray the surface vigorously before I start working.
I'm gonna turn it off for a second and check if there is a sound because I think it's a bit loud and I